Hello, everyone, and welcome to the first EHA meeting on our In Vivo Scribe Corporate Workshop session. We're going to have two excellent speakers that will be joining me on talking about different NGS methods for clinical assessment of hematologic malignancies, both B and T cell clonality, somatic hypermutation assessment, as well as MRD detection. So we are going to be talking about at the beginning some of the experience that I myself have had with routine clonality assessment in a clinical laboratory, both assessment of clonality and using the same assay for monitoring lymphoid malignancies. And then later on, you're going to hear from both Professor Sarah Galimberti and Professor Marcus Tiemann on their experience with also MRD detection and the assessment of CLL and other malignancies and NGS clonality assessment assays. We'll be talking about how we incorporated next generation sequencing methods for routine clonality assessment and disease monitoring. So as you know, assessment of residual disease, both minimal residual disease or measurable residual disease in hematologic malignancies is becoming incredibly important. In itself, residual disease is prognostic and is independent, and this is independent of other clinical and genetic risks. It really serves as the best guidance for additional treatment in terms of intensification, novel therapies, or transplant. As it is at this moment, MRD is being incorporated as part of the guidelines, particularly for acute lymphoblastic leukemia, chronic lymphocytic leukemia, multiple myeloma, and acute myeloid leukemia. And you can do this in several ways. So obviously, the approach is you first have to diagnose the disease. And once you get the diagnosis, you can phenotype the disease and look for the disease later on as you monitor based on the disease-associated immunophenotype. And this is usually done by flow cytometry. An alternate way of doing this is you can look for the genetic profile of the disease and look for the somatic genetic alterations that are particular to this disease, and then you monitor the alterations based on molecular methods, which there are many. The final way that you can do this, and especially obviously for lymphoid malignancies rather than myeloid diseases, you can actually look at the clonal rearrangement that you get for IgH and the T cell receptor, and then you can monitor the patient-specific clone. It is important to mention that residual disease measurement, the amount of information that you can get for your own patient and the risk that they have of relapse fully depends on how deep you're able to do this monitoring. So, for example, if you're only doing this by morphology, there are, as I said, several strategies for doing this. But if you do this by morphology, morphologic assessment is very difficult if it is less than 1 or 2% disease. So patients that remain with persistent disease, morphologic disease, they usually are going to relapse even very fast, even if you do not see morphologic disease or unequivocal morphologic disease. If you use other methods such as flow cytometry, qPCR, digital PCR, or NGS, depending on how you make, how deep you make that assessment, your relapse will depend on how much disease you can actually measure. So if your method is only able to look at a molecule anywhere between 1 in 100 to 1 in 100 or 1 in 1,000, we are more likely to get early relapse. However, if you're able to go to 1 in a million, which is usually what we talk about, minimal residual disease, these patients, if they don't have disease within that depth of assessment, they usually remain disease stable. So for when we're talking about minimal residual disease or measurable residual disease, we're hoping that our methods are going to get us to that depth of assessment to be able to say whether you have one molecule that is disease related in a million, or at the very least 1 in 5 and in 100,000. All right, so there are several challenges to the clinical application of MRD testing. So you can look at this in several ways. As I said, you can look at it by looking at mutations. And as you know, mutations are not necessarily unique or specific to the patient. So it is difficult if you're using KRAS or BRAF or any other marker that happens to be recurrent to be able to do this assessment, because again, if you have highly sensitive methods, you're looking for a specific marker, and the more sensitive the method, you can actually, you have the risk of getting more false positives. Also, there is significant genetic diversity in terms of mutations, and these mutations are not necessarily stable drivers that you can track throughout the disease. Some of them are better than others, but there are some that are not easily trackable. You could also track a lot of these diseases by specific fusions, which may be seen in many of these malignancies, but a problem with the fusions is that fusions are sometimes very difficult to design the assays to be able to pick up these fusions, because there is a lot of promiscuity in the gene partners and variable breakpoints that you can see in this malignancy, in this specific fusion, so that to be able to design an assay that can detect all of the gene partners and all of the variable breakpoints is sometimes difficult. Another thing is that sometimes the clones are not necessarily expressed at every point. So if transiently the disease is not expressing the transcript, sometimes these diseases can evade detection. And finally, you could use surface markers, which is what you usually would do with flow cytometry. But there are some diseases that the antigenic surface marker is actually not there, and sometimes these markers are necessarily universal. Another point is that you do rely on an intact cell surface to be able to pick up this specific epitope. All right, so this brings us to IgH and T cell receptor rearrangements as molecular markers. So both IgH and TCR rearrangements have a distinct appeal as a way of measuring minimal residual disease, and these are the type of markers that are particularly useful for any B or T cell neoplasm that does not contain a chromosomal translocation or a trackable or stable genetic marker. Patients with a specific rearrangement that can be diagnosed at the time of initial diagnosis of the patient usually have clones that are not only unique to the patient, but also are highly stable and highly trackable, unlike recurrent genetic events, which may disappear and appear even if the patient has disease. So just to go over and review very briefly, as you know, both B and T cell receptors undergo a specific physiologic rearrangement, which brings together different parts of the V, D, and J regions. So here is a sketch of IgH and the different parts of the gene. And through through random rearrangement, you end up with different sections of the entire gene that are taken out from the sequence to form a unique sequence that is specific to that B cell. And a similar rearrangement happens for the T cell receptors. These are later on transcribed into a molecule that will be expressed in the surface of the cell, and is absolutely unique to that cell. So any other cell that would have this specific type of rearrangement has to be from that, it has to be coming from that specific cell as a clonal expansion of that specific cell. And additional small changes also happen in the variable regions, which are called through somatic hypermutation, which makes these molecules even more unique. So at the end, the resultant diversity of both IgH and TCR molecules is so high, as you can see, the total diversity after all of these changes is about 1 × 10⁻², which makes them virtually unique to that patient. So it's a very, very easy way to monitor that patient for the disease-specific clone. So clonality testing in general, or this type of assessment, has always been done with sizing assays. For the past 10, 20 years, we have been doing this with assays that are highly standardized and generally look for specific targets in IgH, which are particularly the FR1, FR2, and FR3 regions. And this is an assay that gets done by PCR, where the products are separated by size only. And in here, for example, you can see the products separated by size, and the height, the size is on the x-axis, and the height is the amount of PCR product that you have. And in general, you would have this distribution for a polyclonal population. But if you have a monoclonal population, you would only see one peak. But this type of assay doesn't give you any information on the sequence, and it has no quantitation capability, and it also has a low sensitivity, which is imparted by the fact that many fragments, despite the fact that they have a different sequence, they may have a different size. On the other hand, we have NGS methods, which target similar regions, FR1, FR2, and FR3, but also target the leader portion of the gene. In this case, the products are separated not by the size alone, but also by the unique sequence. And it also allows you to do quantitative comparisons and track individual populations, not only the clonal population that is the malignant one, but also the polyclonal background. There are, of course, marked advantages over length-based analysis, in the sense that, of course, now you have a specific DNA sequence of the clonal rearrangement. And again, not only the sequence that is associated with the disease, but also the entire repertoire, so that you can see increases and decreases as you treat the patient. You can also see clonal events that are hidden in the polyclonal background, because now you have the sequence. You can monitor the dynamic aspects of the malignant clones and the overall immune repertoire at the same time. And of course, as I said, you can make quantitative comparisons. So if you were to plot the different sequences that you would have on a malignancy, you could see that you have different types of clones, for example, and this is one of the analysis that we usually do, where we have one clone or several different clones with different proportions, as you can see in this specific plot. So as I said, the assay, the in vivo scribe assay for IgH, for example, covers the same regions as the fragment analysis does in terms of the targeting with specific primers, the FR1, FR2, and FR3 region, a consensus J primer, and also a leader primer, which allows you to detect more clones than if you used FR1 and FR2 and FR3 alone. So in our laboratory, for example, or in our clinical laboratory, we have a specific workflow that we use for all lymphoid malignancies that come in. In general, we request for each patient to have their tumor sequenced by our MSK impact assay, which looks for genetic mutations in 400 related genes. And these are genes that are associated with hematologic malignancies. But we usually also ask for bone marrow, and we asked for normal controls, which are usually nails or saliva, to be able to sequence and know that all of the mutations that we have are being assessed specifically for somatic alterations and not variants that could be germline. But then at the same time, every single lymphoid malignancy also gets clonality testing. And we usually look for clonality assessment. Our clinicians are hoping for a diagnosis within about 2 days. So we usually screen them by fragment analysis. And if the fragment analysis shows you a positive result, then we go ahead and characterize the clone. This clonal sequence gets saved in a database, along with all of the mutations that we got from the genomic characterization with both DNA and RNA, for fusions, for copy number alterations. And then we, again, save this in a database where we wait then until we get the second sample for monitoring. Samples in general, for clonal rearrangement, as they come into our lab, they get stratified into diagnostic and monitoring samples. For all diagnostic samples that have more than 5% tumor content, these are suitable for clonal rearrangement or for characterization of the clone. When the sample has less than 5% tumor, we usually look for another sample so that we can do a proper characterization. We generally sequence in a staged fashion. So we do FR1 and leader first, and then we do FR2 and FR3 primers. If we get a monitoring sample without a diagnostic sample that has sufficient disease, then this is the type of patient that we wouldn't be able to monitor. But we do try to go to our archives to be able to find samples that are of higher tumor content, or we enrich the sample either by which we can do by flow or by cell sorting. When we do the monitoring samples we usually do the monitoring samples with a much higher input than we would do our characterization samples. And we do them at least in duplicate depending on the amount of disease that they have. This is a characterization sample, for example. And in here, what you can actually see is the output of the top clones. This is a clonal population that actually has 2 different clones with the same V usage. And this is the output from the in vivo scribe software, where you can see the clones arranged by the VJ family usage. We do have our own analysis pipeline, and we actually look at these both by the VJ family or the fragment size, and that's why you see 2 different clones in here compared to here, because they are actually arranged by size. Every time that we sequence a sample, the very first time to diagnose the clone that we're going to track, we do specific QC checks so that we make sure that that clone is unique to the patient. So what you see here, for example, with the QC in green, is the fact that we have actually looked at every single patient sample that we have done in the past and ensure that no other patient has this type of clone as part of our QC checks every time that we do. So what is the success of clonal detection at the time of diagnosis if you use 5% as your cutoff for tumor content? It's actually very good, and it's very close to what you see with fragment analysis. The comparison between fragment analysis and NGS is actually very close. However, clonality by NGS does detect more clones that are hidden in the polyclonal background, and this is particularly more for those cases where you have a prominent peak that you wouldn't be able to diagnose as a clone through a fragment analysis assay, but you can actually very easily see it with next generation sequencing. There are, of course, some samples where you are not able to detect the clone by either assay, and these are usually cases with very high somatic hypermutation, for example. But overall, and this is a study that we did last year, looking at all of the samples that we have run, well, at that time that we had run for characterization, so close to 800. Most of these cases could be easily characterized with only FR1 and leader, and very few cases actually required any more sequencing beyond that to be able to characterize the clone. However, samples that have a lot of somatic hypermutations, such as follicular lymphoma, plasma cell neoplasms, lymphoplasmacytic lymphomas, they do require the use of other of additional sequencing with additional primers, both FR2, FR3, and sometimes kappa, to be able to characterize the clone fully. So once we have the clonal sequence that we're going to sequence, again, we save that in our database, and every time that we get a new sample post-treatment, then we track the clone. This is an example of a monitoring case where clearly by arranging the fragments by size, you're not able to find the sequence. However, because you know the sequence, the specific sequence of that clone, then you can look for it no matter how deep within the polyclonal background this may be hidden. So in this case, for example, the sequence that we were looking for that we characterized at the time of diagnosis was present in this sample in about 0.03% of the total rearranged reads. How well does this work compared to flow cytometry? So it actually depends on the disease. So if we looked at all of the cases that we have done, and this is specific patients that we have done in the past 3 years, if you looked at all of the plasma cell neoplasms, for example, NGS and flow cytometry work almost exactly the same. So in green, what you can see is the cases that are detected by both NGS and flow, and in blue, the ones that are detected by flow only, and in orange, the ones that are detected by NGS only. So for plasma cell neoplasms, both the flow cytometry and the NGS assay really is very much the same. There are a few more cases here that are picked up by flow only, but also a few more cases that get picked up by NGS only. And this is all related to the fact that we do have an excellent flow cytometry protocol for plasma cell neoplasms where we capture more than 5 million cells, and we can get to that 1 in a million mark that we're looking for. This is not necessarily the same for all flow cytometry that gets done across the country. And even in our own institution, the flow cytometry that gets done for some B-cell lymphomas and for B-ALL doesn't actually hit that 1 × 10⁻⁶. But overall, NGS does work better than flow cytometry to be able to detect these populations. For the B-cell lymphomas overall, we actually detect many more by NGS only, and this is anywhere from 10 to 20% more cases compared to flow cytometry, and similar for the B-ALL cases. The correlation with flow, even though we can say we can detect disease or not detect disease, and it's usually the concordancy is actually high, the actual correlation in the level of disease with flow can be highly variable, and it depends on many factors. The fact that, you know, the different markers that are used, the viability of the sample, the surface marker expression, the number of events that are captured, and also how is the data expressed. So an important thing to note for NGS testing is that a lot of the NGS testing, the way that you express the value of the residual disease that you have, is not necessarily the total number of cells that are in the background, but is actually the total number of sequenced amplicons. And as you know, you're only with IgH and T cell receptor analysis, you're only sequencing the gene when it's physiologically rearranged or pathologically rearranged, and not all of the cells in the background are going to have that physiologic arrangement. So many times you're going to have an overestimate of the disease level in situations where you have a low polyclonal background. So if you express the data by the number of index sequence reads, which is the sequence, the reads that are associated with the malignancy, compared to the number of rearranged amplicons or sequenced amplicons, then you're going to end up with a completely different number that you would end up with flow. So this brings us to the importance of normalization and how much of the clone do you actually have. But this is actually important because how much of the clone in reference to the total cells that you have in the background is critical as a dynamic measure of disease burden. And this has been addressed in several ways by different groups. You could use spiking controls to be able to recalculate the overall background number of cells. And you could do this with plasmids that contain a known IGH gene rearrangement. You could have synthetic or known control templates, or you could have even a spike in of defined amounts of buffy code. What we are using at our institution, and this is an example case just to show you, you can incorporate a normalization control. This is the LymphoQuant. And what you would actually do is spike in a very, very small amount of a known sequence so that you will be able to back calculate how much product you actually had. So if we were doing this type of testing, this is a patient that has some disease, residual disease. This is the index clone, and this is the amplification of the spiking control. If you were not looking at a normalized number, we would have an amount of disease that is 8%. And this is 8% of the disease clone over the total rearranged reads. However, the corresponding float is only 0.03%, which is a very, very big gap. And this would give clinicians a high degree of concern because the patient actually has high level of residual disease. However, if you use the spiking control to back calculate that number, the normalized level of disease is 0.07, which is very, very close to what you see here in the corresponding flow. And in general, you do see trends in terms of the amount of disease that you have through the normalization that we're doing with the corresponding flow, but it's actually not perfect, and it depends entirely on the disease. In plasma cell neoplasms, where the flow usually underestimates the amount of plasma cells, our numbers tend to come closer. For this specific patient, for example, if you tracked all of the time points that we have been looking at, you can actually see that the correlation between disease load by flow cytometry and by our normalized number is pretty similar, but it can actually be all over the place again, depending on the disease. So while you do not have a very good correlation between flow, you may not have a very good correlation between flow cytometry and your NGS number. It is actually very highly reproducible, and even reproducible between not in the lab only, but also between laboratories. So for example, this in here is our inter-run reproducibility using our MRD control, which is at 1 × 10⁻⁵, which was run 231 times in different runs and different instruments. And they actually are very tight, and they usually hit that mark. The interlab reproducibility, for example, this is a summary of 57 cases that we run in our lab compared to the same samples run by in vivo scribe, and you can see that the correlation is, again, actually very, very good. So both the inter-run reproducibility and interlab reproducibility is very good, and the correlations with the morphologic disease and the flow cytometry are fully dependent on the type of disease that you have. So there are some very important considerations to point out. If you're using clonality testing with the intent to monitor disease, which is what I think we should be doing, if the intent is to do just clonality testing to say, well, there's a clone or no clone, you could be very easily using fragment analysis as a very rapid and robust type of assay to do. But the beauty of having a clone and having the sequence is that you can actually monitor the sample, and is the intent that we have whenever we monitor, whenever we do clonality testing by NGS. This, as I said, has to be done with samples with high level of disease, and the very, very lowest of disease that we actually would take is that 5% disease. Because otherwise you have a very high risk of ending up with a pseudoclone. The other thing that is important to consider is to make sure that those patients are assessed, if at all possible, at the time that they have their first diagnosis of disease, and not in samples that are post-treatment, because these are usually samples that have very low template and have also reduced repertoire. So it's very likely that you could end up with a pseudoclone. And if you do want to do the testing, or you have to do the testing on a patient that has already been treated, it's important to confirm the clone with more than one primer set to ensure that that clone is stable throughout the different primers. But it's also important to ensure that the clone is, again, unique to the patient, and this is particularly important for T-cell receptors. For T-cell receptors, for example, we like to use both the productive and the unproductive clones, and for diseases in general, just because it gives you higher specificity when you're tracking. So if you have a sample that is biallelic, for example, it's important to track both clones, which gives you an additional level of security when you're tracking. But we did go in our samples post-treatment to see if we were able to actually characterize the same clone and through a blinded study. And so basically taking all of our post-treatment samples, analyzing them as if we were analyzing them for a characterization of a clone, without the knowledge that we had a prior clone that had been characterized at the time of initial disease. And what we do find is that in those samples, if you're not paying attention to the amount of disease that they have, if they have very little disease, and the clone is not dominant enough, you can end up at that low level with a high amount of pseudoclones. So it is those samples that have low level of disease that would be characterized for the first time, where you end up with clones that are not the actual clone that you should be tracking for disease. So that disease characterization really has to be done at the time that they have not been treated, or that they have a lot of disease and not low level between 1% and 5%. So another important factor during your monitoring of the samples is that there are two things that are critically important. Well, obviously, DNA input is entirely critical. If you took the theoretical yield of 6.5 picograms of DNA per cell, to be able to get to a sensitivity of 1 in a million, you need a lot of DNA, about 6.5 to 7 micrograms to be able to get to that level. This is not necessarily feasible in a clinical laboratory, because you would have to have one entire run per patient. So in our lab, we have decided to do 1 times 10 to the minus 5 rather than 1 times 10 to the minus 6. But as you can see, as you saw from the prior slides, this actually gets us to exactly the same level of performance as the flow cytometry, even though the flow cytometry is getting to 1 times 10 to the minus 6, and this is because of the specificity of the clone that you're looking for. But another thing is that the workflows when you're doing minimal residual disease testing in a laboratory is incredibly important to redesign your workflows. You have to have a rotating schedule for the sequencers, and also strict washing procedures to ensure that one clone doesn't carry over to another run. And also to make sure that you divide your characterization samples and your monitoring samples. So every time that you have a characterization sample from a patient, you should not be running the staging sample in the same run to ensure that you don't have barcode hopping or barcode contamination. In terms of validation and implementation, so this is something that to be able to do this in a laboratory, it has to basically be done with not too much guidance. So in our laboratory, we had to do this based on publications that are there, that have used many different methods for their clonality testing and minimal residual disease assessment and monitoring. But the guidelines are not yet out to be able to know exactly how to do this. And not too many laboratories are using this as routine clinical care. So their experience is not widely available. There is also a very heterogeneous type of assay design between institutions and protocols and data interpretation. So it's actually difficult to know exactly how to go forward with these validations, but the guidelines are being prepared for both Europe and the United States, hoping that we will be able to do this very soon. But the fact that right now there is a lot of proprietary technology that is being used really precludes using that type of data from as a meaningful guide for a clinical validation or broad adoption into routine clinical practice. So in conclusion, I think that in general, routine IGH and TCR rearrangement assessment for both clonality and for minimal residual disease is very possible and very feasible in any clinical laboratory, provided that the workflows are organized correctly and also that they are validated fully. Compared to the fragment analysis assays, they do have much higher sensitivity for characterization. But if you're going to use this for monitoring, I would not push the limits for that characterization, even though it does have higher sensitivity, and try to keep with a high tumor content type of sample to be able to do your first characterization if the intent is to use it for monitoring. But when you do use it for monitoring, compared to flow cytometry, they do have a much higher detection for residual disease in both B and T cell neoplasms. This is not, in our experience, necessarily the same for the plasma cell neoplasms, but it has a lot to do, again, with the high sensitivity protocol that we have for plasma cell neoplasm flow. To successfully implement this, it really does require new standardization and validation and implementation processes, which really should be performed as a multi-center and multidisciplinary collaboration, which are already undergoing and are not yet published, but they're definitely underway.